Hello everyone, this is the second of two videos that I'm preparing as preliminary videos for this class. This class will primarily focus on data mining using the JASP package. And if you've read or looked at the other videos, you'll know that already. But what I want to be able to cover in this video is something that we call inferential methods or inferential statistics. And in the first video, if you've seen it, you saw that I talked a little bit about the process of looking at samples in order to make draw conclusions that we could then generalize to populations. We're going to go into a little bit more detail here. This is going to cover a little bit of the scientific method, but it's really meant to sort of introduce you to concepts that you're likely to run into in the JASP package so that they make sense to you when you see them. So there are two types of statistics. We've already run into one if you've watched the other video. And that's descriptive. In descriptive, we're using tools that basically descriptive statistics describes a data set. And it gives us the ability to learn things about what's in the data set. That's particularly useful when we start doing data mining, because the data mining process is trying to find interesting things in the data set. But this will just give us, descriptive will just give us a general feel for what's in that data set. It includes tools like mean, variance, standard deviation, median, mode, and range. And again, we're looking at a data set and we're trying to figure out things about a data set. There are many other tools that we can use to learn about the data set, but these are the most common. Inferential is the process of kind of dealing with the reality of large data sets. And when we're dealing with large data sets, we may not have the ability to learn very specific things about large data sets just because they're too large. So what we do instead in inferential methods is we take a representative sample. A representative sample is a sample that pretty much reflects the population, the overall large population, in many different aspects. And generally, a good rule of thumb is that every member of the population has an equal chance of becoming a member of a representative sample, which makes it a true representative sample. So we select a representative sample that looks in many ways like the population, and we conduct our tests on that smaller sample, which becomes realistic because now we're dealing with 100 or 1,000 records as opposed to 10,000 or 1,000,000 records. And what we infer from the test on the sample is something that we can generalize or sort of infer that is actually happening in the population. So if it's one way in the sample and we drew the sample as a truly representative sample, then it will be that way in the population as well. And that's what we're going to talk about. So representative sample is really important. It's really important that you pick a good one. So like I already indicated, every member of the population has an equal chance of being in the sample. So the sample demographics reflect the population demographics. So the same percentage of males versus females, of older versus younger, that the distribution of different demographics is the same percentage in the population is the same percentage that's in the sample. The sample must be large enough to produce meaningful results. A sample size of 10 typically is going to be too small. There's too much opportunity for bias. There's not enough opportunity for variance amongst the sample, so it's not going to be useful. A sample typically has to be at least 30, sometimes higher. It depends on the level of confidence that you want to have. But the larger your sample, the better your results are going to be, the more generalizable they will be back to the population. And we also want to make sure that there are no confounding factors that will impact the results. So I used an example of wanting to learn something about the US population that is over 50, but my sample only has people between the age of 50 and 55. The sample is too different from the US population of people over 50. It's not going to give me useful results. But there could be other confounding factors that are going to impact the results, and we may talk about them later in this presentation. Here's an example. Political polls typically use around 1,000 respondents to represent all voters. And what they want to do is they want to make sure that the 1,000 respondents that they select, everyone has an equal opportunity to become one of those 1,000, or all voters have an equal opportunity to become one of those 1,000, and that they represent the US population in terms of demographics for race, culture, gender, and those other things. That way they can get an accurate result in the population that they can generalize back to in the sample, that they can generalize back to the population. Bias are factors that make the sample different in the population, and I just gave you a great one with the age of the sample members. But here's another one for political polls. Some political pollsters, and they may have gotten away from this now, but they were phoning respondents, and typically younger respondents who tend to be more liberal politically would not answer the calls. They would filter the calls out, or they would take them on their cell phone or their mobile. So it was the older folks that were answering the calls at home that were responding to the polls, and that was skewing the responses more towards the more conservative views of the older respondents. So using the representative sample, it's separate from the population, and we're going to learn things about our sample. If the sample's good, what we can learn can be generalized to the population. Quite often, we are interested in the relationship between variables or the impact that one variable has on another. So this could be the relationship between hours studied and score on a standardized exam. Those two variables are both numeric variables, and we are interested in the relationship that they have with each other. Our work is to determine if the relationship or impact is significant in the sample. If it's significant in the sample, then we might be able to sort of infer that it's significant in the population. And significance has a specific meaning. Oftentimes we throw around the word significance to be something that's important, but in statistics, significance has a very specific meaning in the sense that we are given a result that has we have enough confidence in it that we determine it to be statistically significant. So here's how this works. We start with a question, and I will use this example. Do first-generation college students perform better academically than non-first-generation students? So first-generation students are students who are the first ones in their families to go to college. And non-first-generation students are not the first ones in their families to go to college. Maybe they had a parent that finished college, or several members of the family have already preceded them through college. So our question is, do first-generation college students perform better academically than non-first-generation students? So the next thing we do is we identify variables that represent the items in our question. So what is first generation? How do we determine first generation status? That is generally recorded when a student applies to a university. That's one of the questions on the application process. And enough students will respond to that question that we can get a good sample of first generation students. We would also get the opposite, non-first generational students, because enough of those apply to say, no, I'm not the first person to go to college. And we might pick a measure of academic performance. That could be a GPA. A GPA is a poor measure of academic performance. While it might be great that a person has a 4.0 GPA, there are too many confounding factors that impact the calculation of a GPA to make it not a consistent measure of academic performance. But for convenience sake, we'll use it here. We formulate a hypothesis or hypothesis pairs. And typically hypothesis pairs are opposite statements of each other. We have one that's called the null hypothesis, and that's designated with a capital H and a subtext 0. That's generally the terminology that we use to say this is the null hypothesis. And with the null hypothesis, we are stating that there's no difference or there's no impact or one variable is not related to the other variable. In our example, we could say that null hypothesis that there's no difference between first generation college students' performance or GPAs and non-first generation students. That is our null hypothesis, that there is no impact, there is no difference. The alternative hypothesis, quite often referred to as research hypothesis, is designated as H subtext 1, and that's the statement of what we're really trying to find out. We are going to state that there is a statistically significant difference between the performance of first-generation college students and the performance of non-first-generation college students in regards to their GPA. So now we're set up. The null says nope, and the alternative says yep, that there is a difference. So now we conduct our test. When we conduct our test, the focus of our test initially is on the null hypothesis, because the null hypothesis is an opposite statement of the hypothesis. So we want to know, can we reject the null hypothesis? Remember, we said there is no difference. So can we run a test that would allow us to say, wait a minute, there is a statistically significant difference, and can we reject that null hypothesis? If we can reject the null hypothesis, that leaves us with no alternative but to accept the alternative hypothesis. So we would then have support for our hypothesis that non-first generation college students and first-generation college students have statistically significant differences in their performance as shown in their GPAs. So how do we know? Well, determining whether or not we can reject the null hypothesis is really kind of a bit of a call that is made by the researcher. And we use something called a p-value. A p-value is returned with every statistical test. And the p-value in the simplest forms, and I'm sure I'm going to talk about this on a later slide, is how confident are we in the results that we got back from our statistical test. So what is the p-value? It's a probability. It's the probability, and it ranges from 0 to 1. It's the probability that your results that you got in your statistical test were purely by chance, that they have no statistical significance, that there's what's the chance that what you're showing in your statistical test is just meaningless. Okay? It takes into account the size of your sample. So the larger the sample, the more likely and more confident your results are going to be. It looks at the nature of your statistical test. It could be I'm losing the term here. Correlation, that's what I wanted to use. It could be correlation, it could be ANOVA, it could be some other test. So we'll look at the nature of your test. It looks at the values of your measurements. Are they varied? Is there a lot of variance in the values of your two variables? Are they very aligned? Do they look very similar? Every statistical test has p-values, as I've indicated, and you want your p-value to be low. Because remember, p-value is the chance. What's the chance that your results are not meaningful? And so you want that to be low because you want that chance to be very small. So a statistical test will generally give you two things, usually more. It will give you the results of your test, and it will give you a p-value. Now, that is a very broad statement because most statistical tests will give you many, many more things than that. But for now, we're going to limit our focus just on the results of the test and the p-value. The p-value will tell you the significance of your results. So results with a very small p-value, those are good results. And results with a very large p-value, those are not so good results. They're not valuable. Keep in mind this when you're in regards to p-value. P-value, again, is the chance that your results are not meaningful. The chance that your results are garbage. So when you have a small p-value, that chance is very small. But you still have a chance. There's still a chance that your results are no good, that your results that you arrived at were just purely by chance and they're not meaningful at all. We want to minimize. We're never going to get rid of that chance that the results we got are not good, but we want to minimize it. So we want to lower the probability, and that's why we want the small p-values. There has to be a point at which we decide, you know what, I'm going to accept a 5% chance. I will be willing to say that if I've got 95% chance that my results are good and a 5% chance that my results are meaningful, or not meaningful, then that's good enough for me. Because it's going to be 19 times out of 20 that my results are going to be good. Sometimes it's going to have to be even lower than that. I'm going to want to see, I don't want any more than 1% chance that my results are not good. So that's a much more stringent test. It's a much more difficult result to achieve, but many researchers set their p-values to be 1%. So in short, the p-value is the likelihood that result you got from your test were pure chance and have no statistical significance. A small p-value, there's a very small chance that your results are pure chance. And a large p-value, there's a much better chance that your results are purely by chance. So what do p-values look like? Well, sometimes they look like p equals 0.000254. That's a pretty good p-value because that means even if you adjust that's a decimal, if you adjust it to percentages, you're going to have about a 2.5 hundredths of a percent that your results are not useful. Another p-value could be 0.316. That one's not such a good p-value because if you turn that into a decimal, it's going to be a 31.6% chance that your results are purely by chance and are not meaningful. So that's not good either. That's just not good. More often, we often see a p-value that will look like this. P equals 0.045. Change that one to a decimal and you have a 4.5% chance that your results are not meaningful. It depends on the threshold that you set. You have to say, well, okay, that's a 4.5% threshold, or 4.5% chance that my results aren't meaningful. I'm willing to accept 5% or lower that my results are not meaningful. So that one's okay. So the first one is okay. This third one is okay, but the second one is not. So roughly translated, you see that you have a 0.0254% chance that your results are pure chance in the first example, a 31.6% chance that your results are pure chance in the second, and a 4.5% chance that your results are pure chance in the third. But what, as you as the researcher, what are you willing to accept? So you could say I'm willing to accept a 5% chance that the results I'm looking at are pure chance. That's a 95% chance that they are significant and meaningful. And that's the threshold that you're going to set. I want to say that my p-value is going to have to be less than or equal to 0.05. If I'm only willing to accept a 1% chance that the results I'm looking at are pure chance, and a 99% chance that they are significant and meaningful, I may set my p-value threshold to be 0.01. The most common p-value threshold that you're going to see in research, whether it be social science or other research, is going to be 0.05. Quite often, however, in scientific research, where there needs to be much more precision, you'll see p-values set to 0.01. So let's go back to the hypothesis. Remember, the null hypothesis said that there was no difference between first-generation GPAs and non-first-generation GPAs. The alternative said that there is a difference. The hypothesis pairs are always opposite statements of each other. So now we're going to gather the GPAs of the first-generation students, and we're going to gather the GPAs of the non-first-generation students, and we'll compare them using a t-test. We'll learn more about t-tests later in the class, but a t-test basically allows you to compare two groups to determine if there's a statistically significant difference in the mean values of those two groups. So essentially what we're doing is we're taking all the GPAs for the first generation students, and we're coming up with an average. Then we're going to take all the GPAs for the non-first generation students, and we're going to come up with an average for that too. If those two averages are different, that doesn't really tell us much because we need to understand more about the scatter around those means. So we want to know if they're truly statistically significantly different, and that's what the t-test will give us. When we have two samples, it will look at the mean, it will look at the scatter around the mean, and tell us if there's a significant difference between the two groups. So when we run a t-test, we get two things. We get the t-statistic, which is our test result, and we get a p-value. We decided that the p-value threshold that we wanted was to make sure that any p-value we got was less than 0.05, and that would help us determine if our test result is significant. So here's an example. Say we came up with a t-statistic of 2.51 and a p-value of 0.00214. 2.51 is indicative of there being a significant difference. We'll determine how we can determine that, but a significant difference between our two groups. So what we're basically seeing here is that with a t-statistic of 2.51, that becomes a statistically significant result. However, if our p-value was too high, then we'd throw the whole thing away because we couldn't believe it. But in this particular case, with a t-statistic of 2.51 and a p-value of 0.00214, well below our threshold, we have a significant result. So there is a statistically significant difference between those two groups. So now what? Well, our null hypothesis said there was no difference, but there is a difference. So now we reject the null hypothesis, okay? And that leaves us only with the alternative or research hypothesis. So we are able to accept that research hypothesis at our p-value threshold. But what if our t-statistic was still 2.51 and our p-value was 0.067? 0.067 is just a little higher than our threshold of 0.05. So that exceeds our threshold. So we fail to reject the null hypothesis because there's too much likelihood that our results were pure chance, that they're not meaningful. Keep in mind, and this is important, we can reject a null hypothesis, but we can never accept a null hypothesis. When we reject a null hypothesis, all we are saying is that there's too much evidence for us to say the null hypothesis is true. But we can never accept a null hypothesis if the p-value is too high, because we don't have any proof that it's true, that it's correct. So just because we can't reject it does not mean we have to accept it. In this particular case, the test that we ran showed us that there was in fact a difference between the two groups. But what if the test that we ran told us that showed us that there was not a difference between the two groups? It doesn't mean that we have any proof that there really isn't a difference. It just means that we have not enough confidence in our test to say that there is a difference. I know that's a subtle difference, and I know it's a bit confusing, and probably not 100% important that you remember it for what we're going to be doing in JASP. But just know, if you remember anything out of this conversation, you can reject a null hypothesis, but you can never accept a null hypothesis. Okay. So here's another example. Imagine we have a list of test scores. One group studied using new study guides that were prepared by the faculty. And the other group studied on their own. So the null hypothesis is the study guides made no difference in the test scores. The alternative hypothesis is the use of the study guide led to higher scores. So we're also going to use a t-test on this one. We're going to compare the two groups. Once they've taken the test, we'll compare their test scores. We'll calculate the mean, and we'll use the distribution around the mean in our t-test to determine a t-statistic and a p-value. And before we do this, we're going to set our p-value threshold to less than 0.05. So what if the t-stat is 2.831 and the p-value is 0.0234? Or what if the t-stat is 2.831 and the p-value is 0.234? Well, in the second example, we're going to reject the results, but the results are not useful, and we're still stuck with the null. Remember, we can't accept the null. We just can't reject the null. We can't be confident that the null is wrong because our p-value is too high. In the first one, we have a potentially useful t-statistic at 2.831, and we're confident in the results of the test because it is lower than our p-value threshold that we've set of 0.05. What we don't know yet, though, is 2.831, if that is a useful result on a t-test. I mean, just looking at it, you can't just say, well, yeah, it's a result, but is it high enough? Is it good enough? We don't know yet. So all we know is that it qualifies on the p-value side as being lower than our threshold. But we don't know about the t-statistic, if that's important or not. We'll talk about that coming up here. The t-statistic in the t-test is determined, as all statistics are in many statistical tests, driven by a couple things. One, the size of the groups. So the larger the group, the better, because we have more data to work with. But we also are going to be using something called critical values. In the back of every statistics book are tables of critical values. And critical values are where we're going to go to determine if our t-statistic, which we've already qualified because the p-value was low, but if the t-statistic is significant enough or high enough to suggest that there truly is a difference between those two groups. So each test has its own group of of critical values. There's one for t-tests, there's one for ANOVAs, there's one for many different tests. And it will tell you if your result is meaningful, right? We can say that something is statistically significant, but we can also say that it's meaningful if the result we got from the statistical test is higher than the critical values. We could have alternatively, we could have a result where the result is statistically significant, but the difference between the groups isn't high enough to make it meaningful. And that also is determined by the values that are in the tables of critical values. So in order to check to see if the value that we get is useful, we need to know the size of the groups, and we also need to know the degrees of freedom. Each test has its own table, as I indicated. We use t-tests, so we'll use the critical values for the t-distribution, what they call the t-distribution. The degrees of freedom are closely related to the size of your groups. They're generally just slightly smaller. So you take the number of, like for example, if we had 30 students in our first gen group, or well, they were moved on to the test scores, didn't we? Have 30 with the study guides and 30 without the study guides. We would have 60 total. That would be driving the degrees of freedom and how we would use that with our groups, with our tests. We also need to know the p-value threshold, so we'll leave that at 0.05. So assuming we meet the p-value threshold, we look at the table, the back of our statistics book, or online, you can find them online as well, to see if our result means anything. And the table shows us the critical value that we must meet or exceed for the result to have any real significance. Here's an example of critical values. So imagine that our group had 20. And for the t-test, we might use, and this is determined by your test. So every test is different, but it's essentially n minus 1 or 19. The reason why we shave one off is because there's always going to be one example, one test, one combination, one comparison that is static, that will never change, never move. So look at your statistical tests, and we'll look at these when we get into JASP, and it will help us determine the degrees of freedom. But with there being one example, one instance where there really is no freedom, the values can't change, they can't move, we don't get to count that one. So we basically are counting only 19 when the number of n was actually 20. So the degrees of freedom are important. Then you see that if the p-value threshold is set to 0.2 or 0.1, 0.05, 0.02, or 0.1, those are the critical values that we must meet or exceed to determine if our test is meaningful. So in this particular example, we're going to use a degrees of freedom of 19. Our threshold is 0.05, so we'll look at that column. Our t-statistic has to be above 2.093 for it to be meaningful. So a t-stat of 0.831 with a p-value of 0.0234 is significant, but it's not meaningful because for the row, degrees of freedom 19, the column, 0.05, it does not exceed that critical value. It's an accurate result, but we don't care. But a t-statistic of 2.831 with a p-value of 0.234 is significant and meaningful. It passes both tests, and we can reject a null hypothesis and accept the alternative. So yes, it's possible to have a statistically significant test that is useless. So to infer, the null hypothesis, in the sample there is no difference in GPAs for first gen and non-first gen. That was the example that we used. We had a sample of 30 students in each group. We stated an alternative hypothesis, which is that there is a statistically significant difference in the GPAs between the groups. The t-statistic was 2.342, and the p-value was 0.00251. So we have statistical significance. That's test number one. Now we have to check the t-distribution and determine if the critical value, what the critical value is when we're using a p-value threshold of 0.05 and 29 degrees of freedom. And we determined that that critical value is 2.045. We looked it up in the table. We opened up the t-distribution table. We pulled down to the row that is 29 degrees of freedom. We moved over to the column that is 0.05, and we noted the value there, which was 2.045. Our t-statistic exceeds 2.045. So that means our test was both significant and meaningful. So now we can go back to our hypotheses, and we can reject the null and accept the alternative. And with these results in the sample, we can then infer similar results in the population. This is a process of generalization. We have done a really good job of selecting a sample. We have set up hypotheses. We have run a test. We have determined statistical significance and meaningfulness. And now we've rejected the null, and now we're able to infer those results and generalize them back to the population. That's the process of inferential statistics. It's just it's just with a lot of different tests. The process is basically the same. It's just which test you use and which one you employ in order to get to your statistical significance and meaningfulness. All right, that's the second video. Let your instructor know if you have any questions about these concepts. We'll probably, in the next video, be looking at concepts in JASP.